Good evening, everyone. Welcome. Welcome to the Fralin Biomedical Research Institute Maurice Strauss Distinguished Public Lecture. I'd like to, as always, acknowledge the memory of Maurice Strauss and his family, son Steve Strauss, who is here, for all they have done to make this possible. We did a little count, Steve, I haven't told you yet. Turns out this program has sponsored 70 speakers so far over the years. And thank you so much to the whole Maurice Strauss family. And world-renowned scientists, physicians, thought leaders, et cetera. It's been a real pleasure to have the support from Maury and the Strauss family to bring this program here to everybody in Roanoke. Before I introduce this evening's speaker, I'll do a plug for the next speaker coming up. Our scheduling got a little tight, and normally these are spaced out a month apart, one per month, but our speaker for next month, for November, has to come next week. So it's really back-to-back. So a week from this evening, our next speaker will be Dr. Teresa Lamb. Dr. Lamb is the head of vaccine immunology and professor of vaccinology and immunology at Oxford University in the United Kingdom. She's an honorary officer of the Order of the British Empire, and she's the immunologist who co-designed the COVID-19 vaccine that was administered to millions of people worldwide. And she will speak about pandemics, the lessons I've learned. I promise you it will be an interesting talk. So for those of you who have another Thursday evening available and want to get some more great food and hear a great talk, please come back out again. Okay, so it is really an honor and a pleasure to introduce this evening's speaker, Dr. Jennifer Munson, and the Munson family, who are here, by the way, a few of them. So hey, before getting into details, I want to say the reason we can do this, and I'm not talking about the support of the program from the Strauss family, I'm talking about have people doing this incredible work that you're going to hear about tonight and you've heard about before in this series, is due to all of you, to a large degree, the taxpayers who support medical research, particularly through the National Institutes of Health. And during a time of great challenge, I just want to remind everybody, the reason we're able to do the things we do and the people that you'll hear from are able to do them is because of that support through a highly competitive process. It's not a giveaway, it's very competitive, and the best scientists are selected to do the best science. So we benefit very much in this country and hope that support will go forward. So now I do want to introduce Dr. Jennifer Munson. Jenny, if you don't mind me calling you Jenny, since I do every day, is professor and director of the Fralin Biomedical Research Institute Cancer Research Center here in Roanoke. We actually have two cancer research centers from the FBRI, one based here that Jenny directs and one based up in Washington, D.C. as well. She's also a professor in the Department of Biomedical Engineering and Mechanics. Are we still saying mechanics? Until January, okay. As of January, she'll be professor of biomedical engineering. Now it's mechanics in the College of Engineering at Virginia Tech. Dr. Munson did her undergraduate work in chemical engineering and neuroscience at Tulane University in New Orleans. She was a Fulbright Fellow, went to the Swiss Federal Institute of Technology in Lausanne, did some work there, and she also worked as an engineer in biotech, working at Genentech for a while. So didn't follow the traditional academic route of one degree to another, did other things in the real world for a while as well. And then after that, she went on to do her Ph.D. in bioengineering at Georgia Tech. And she then did a postdoctoral fellowship back to the Swiss Federal Institute of Technology in Lausanne. She joined the faculty up the road at the University of Virginia, you know, the place with the football team, you've all heard about that, in biomedical engineering. And then she moved to Virginia Tech to join the Department of Biomedical Engineering and Mechanics and became an associate professor, I guess, at the time, and then a professor. And then several years ago, relocated her research operation here to Roanoke as part of the Fralin Biomedical Research Institute, where she serves, as I said, as the director of our cancer research program. Dr. Munson has received lots of awards recognizing her innovation that works at the interfaces of engineering, computation, modeling, mathematics, and what we call wet biology, if you will, actually doing experiments and driving them through thought processes that require modeling and very quantitative approaches, as you'd expect from a biomedical engineer. In recognition of her achievements, she's received a lot of awards. I name a couple. Several years ago, she received the Emerging Leader Award from the Ivy Foundation. And more recently, she's been named as a fellow of the American Institute of Biological and Medical Engineering and a fellow of the Biomedical Engineering Society as well. And those are both very competitive and prestigious organizations to be named in. Her work is very consistently followed one particular area of focus, which is brain cancer, what are called gliomas. And even her early graduate work, postdoctoral work, she was working on adjuvant therapies to enhance chemotherapeutic efficacy against these gliomas. She then went on to identify new small molecules and non-invasive compounds to treat them, and then started really getting into what you may have seen from the family demo out there with little ducks going around, this idea of fluid flow and how important it is, the fact that things are moving in our bodies all the time, in our brain, different types of fluids. And as an engineer, she grasped that wholeheartedly, modeled it, has actually taken it to the laboratory to begin to apply it to understand, in what I call a precision medicine type approach, how it can inform treatments of treating things like brain cancer. She's used magnetic resonance imaging, or MRI, to map this flow, this interstitial flow around tumors in the brain, for example. She's also been a leader in something that's happening nationwide, certainly, if not worldwide now, to begin to get us to think out of the box of our traditional models of disease and work more in systems where we can isolate cells and use modeling. And the federal government is interested in funding this, and Jenny's led discussions on this at the national level as well. And this has taken her into the world of immunology, and she now has gone beyond, although she's still doing a lot of fantastic work on brain tumors and glioma. But recently, she had a study out that got a lot of well-deserved coverage on what happens with respect to flow coming from other systems called the lymphatic system when you're exposed to chemotherapy as a cancer patient. Some of you have heard about this so-called chemo brain that people experience during this, and can use her tools to begin to understand that, and even gone into the world of neurodegenerative disease like Alzheimer's disease as well. So I think you can see how the quintessential combination of computation, mathematics, engineering, and biology come together through a brilliant mind to really advance this field. So please join me in welcoming Dr. Jenny Munson.
Thanks, Mike. Can we turn the lights down a little? Because I do have a lot of pictures. That was a really nice introduction. And also, he actually followed the progression of my presentation exactly, and he didn't see it in that summary at the end. So you got a little bit of a highlight reel. So yeah, so it's a real honor to be here and be able to present, in Roanoke in this lecture that, I've attended and watched amazing leaders from around the world come and lecture in. And hopefully everyone learns a little bit and, feel free to ask questions. But I'm hoping to kind of convince you that thinking about fluid flow is important. That's the main takeaway, and give you some thoughts about how engineers can really study this specifically and how it's kind of a unique thing to study. So as Mike kind of mentioned, so my lab does a lot of things. We build a lot of tools. So I'm an engineer by training. So our primary thing, and actually we did an outreach event a couple weeks ago, and every student we asked, what does an engineer do? They said, you build things. And it's like, yes, we build things. And so that's what we do. So, and we build tools to measure, model, and manipulate fluid flow. So we can see it, so we can study it, and so we can fix it when we need to. And so today, during this presentation, I'll show a lot of these tools. I'm not going to go into a lot of detail with some of them, but hopefully you can appreciate, kind of where these things might show up throughout the talk. But why study fluid flow? And specifically today, why study fluid flow in the brain? So fluid flow and fluid in general is vital for your brain function. So it bathes and protects all the tissues that are there. So it insulates things from impact, or if you have an infection or something like that. It circulates and delivers key nutrients. So the fluid in your brain is constantly moving. And it also washes out waste and then replenishes vital nutrients. And it actually maintains this balance in your brain so that you can have, normal function. And actually your brain kind of replenishes about a ½ liter of fluid a day. It makes that and drains that, and that's about a water bottle. So every day your brain is making about a water bottle's worth of water. And so this is what it kind of looks like. So this is not going to have the fluid moving in it, but if we think about a brain, it has these big fluid-filled ventricles in the middle. So that's the green and kind of the orange. And there's a lot of fluid moving in there in kind of empty spaces. And then there's going to be fluid moving through the brain kind of along these kind of spindly things that you're seeing, and that's going to be moving the fluid from the bottom and up to the top, and then eventually draining it out. And so all that is happening constantly. But then if we really kind of zoom in really close, where I study things is in the interstitial spaces. So these are in these like small spaces at kind of a tissue level. You can think like a millimeter or so, about, very, very small sizes, where you have kind of these building blocks of biology, right? The cells that are going to be kind of distributed within some sort of matrix, and they're going to be having this flow moving through them all the time. And that flow is going to interact with them on a regular basis. And so an analogy that I kind of use is if you think about looking in a creek bed and you look right at the bottom and you have all these little rocks and pebbles and sand, the flow is moving in those spaces, and that's what's happening in your brain. But hopefully you don't have any rocks in your brain. And instead it's moving through these cells, and it's much softer. And so that's really what we study. And this is what that might look like inside of a brain. So here we're looking at a brain, and the green is actually the fluid moving through the brain. And it's kind of moving around the vessels, where we have flow moving through the vessels, but it's moving around the vessels and in these spaces. And it does really space fill at kind of this magnification because those cells are so small. And so, that's constantly happening, but it's not always constant. So there's a lot of things that we do in our daily lives that can affect the change the fluid flow in our brains. These include exercise and sleep, which will both increase flow in your brain. You might have those experiences of, feeling a bit more clear-headed after exercising, or maybe, feeling more refreshed after sleeping. And that could be partially because you're getting some of this fluid flow and waste removal from your brain. And then as we age, our fluid flow actually slows down and we make less fluid in our brains. And we don't fully understand what the implications are that, but we're really studying that in my lab as well. And there's a lot of other things that can affect fluid flow. Honestly, every couple days, there's a new report about something that's going to contribute to that. But it's really changing, and it's really specific to each individual person. And then where my lab is really looking at things is in disease states. And so we're going to talk about two today. So one is that we're going to talk about malignant brain tumors, and specifically we're going to talk about a tumor called glioblastoma. And in this scenario, flow is too fast. And then we're going to switch, and we're going to slow it down, and we're going to talk about cognitive disruptions or issues with memory specifically. And in those scenarios, we actually have slower flows. And we'll talk about kind of what those two things look like and how we study that. And so first, in a brain with cancer, we're going to have more flow than we're supposed to in this tissue. And so what is this brain cancer that we study? So glioblastoma is the deadliest brain cancer, and it affects 3 in every 100,000 annually in the US, and its incidence increases with age. The average survival time is 13 months from diagnosis, even with aggressive standard of care. And one of the hallmarks of it is that these tumor cells invade individually into the brain. And so what's happening when a tumor grows in a tissue, and this is true in any tissue, but here shown in the brain, is that you have this tumor, and so you have unchecked, like bad cells growing inside of a confined space. And when you do that, it's going to increase the pressure of that tissue. And so you're going to have a higher pressure in the tumor, but that's still going to be next to kind of a relatively normal pressure in the surrounding tissue. And so if we think about a weather system, if you have a high pressure system next to a low pressure system, you get wind, you get flow moving from the high pressure to the low pressure. And the same thing happens with tumors, is that you have the high pressure resulting in fluid flow moving into the normal brain. And that's bad enough, as you can imagine, that you don't want stuff from a cancerous tumor moving into the surrounding healthy tissue that you need to function. But it's also moving across these zones around the tumor where you have these invading cells. And these invading cells are really one of the primary reasons that this disease is so deadly. And why is that? So if we just think about, there's a whole bunch of reasons, but one of the big reasons is that if we even think about the standard of care and how we treat all tumors, and works in many, many tumors, is that when a patient comes in with glioblastoma, they would come in with some sort of symptom, that's affecting them, then an MRI is performed, and then they receive a resective surgery where the goal is to remove as much of the tumor as possible. So you can imagine if the cells have invaded away from that primary site, it's going to be really hard to get all of the tumor. And then that's followed up with chemotherapy as well as radiation therapy. And radiation, both of those require access to those invading cells in order to kill them. And so as you can imagine, all of that kind of makes these tumors incredibly difficult to treat, and also makes it really important to understand why they're invading, how we can stop them, and then, what does that all mean in terms of treatment. And so kind of going back, we're looking at the flow across this invasive zone. And so we are really trying to understand this interaction between flow, invading cells, and kind of the tumor microenvironment, or the environment in which the tumor is. And so one of the major hypotheses that my lab has been studying for the last 10 to 15 years is that increased flow leads to brain tumor invasion. And so we've been trying to study this. It's not easy to study though, and in order to do this, we have to develop new tools. So we can kind of see this connection, this correlation, inside of a tumor, but in order to test it, we have to be able to very specifically apply flow or not apply flow. You can't shut down flow in a brain, it's not good. So we have to do that in a dish. And so what we do is we design these tissue engineered models or tumors in a dish. And so during tumor resection, you're able to get cells from the tumor and then culture them outside of the body. And we do that in the cell culture hood. And then we also couple that with analyzing patient tissue so that we can make sure that we're getting the right support network, including cells and the matrix that we need to make a 3D system. And then we put those cells inside of what's called a hydrogel. And you can think of a hydrogel as kind of like Jell-O, except you wouldn't eat it. And so in this system, we can actually put these tumor cells, and now we have an interstitial space that we're able to manipulate. And so this is what they look like. So this is just one example. So they're pretty small. And then the pink is actually the kind of tumor in a dish. And then we can apply flow through that gel, and then we can see how many cells have invaded. And so this is all done in vitro, or using cells outside of the body. And so this is what we've found. And so I actually had this huge graph, and it's now simplified into a single column. And so basically, we know that, so here we're looking at how much change, so the fold change with flow. So this is, double the amount of invasion, triple the amount of invasion. And if it's the same as static, then it's going to be 1. And what we see is across a number of different patient lines is that most of them, almost all of them, increase their invasion when you apply flow. Though we do have a few that don't, and we have some that are right near the middle, and those are really interesting, and we're trying to study those. But majority of them really invade more with flow. And then we're able to do things like image these cells inside of the hydrogel and apply flow. And we can kind of see that they start to create these migratory paths that move in that direction as well. So now we have more invasion, and we're also seeing some evidence of directional invasion. And so why is that happening? So one of the things that we've been exploring for a long time and that we've studied is something called autologous chemotaxis, which is a big mouthful, but I'm going to try to explain it. And so basically, if you kind of put yourself maybe outside and say you're carrying like a stack of papers, and it's a really muggy, still day, and you drop the papers, where are they going to land? At your feet? Not at your feet. At your feet, right? And so to pick them up, do you have to take any steps, or can you just bend down and get them? Bend down and get them, right? Okay, now say you're walking on the drill field in Blacksburg, and you've got wind at your back, and you also drop the papers. So where are they going to go? All in front of you, right? And so now, in order to pick them up, you have to start taking steps, right? And so that's what the cells are doing. So basically, these cells are making proteins that they want to follow. And then they're getting pushed up in front of the cell, and then they want to follow them. And so this is kind of the idea, and this is something that we have seen in tumor cells, but it's actually more common in things like immune cells that need to move around your body really quickly to get to a site of infection. And so this is a mechanism by which, kind of goes across a couple different models, but we've been really exploring this in terms of cancer cells. And this really results in kind of this directional migration, which is so dangerous. And so we have a few mechanisms and things that we're studying for that, but it's just kind of an interesting idea of how these cells are actually migrating away from the tumor cells. And so, over and over again, and I'm kind of highlighting there's probably even more people, every undergrad in the lab, everybody in the lab has run the same assay where we're trying to see how flow increases invasion. And so we're just seeing that that's going to happen with a number of our glioma lines and with most patient cells. And so, but that's a really hard problem to solve because you have to figure out which drug would work with which patient cells in order to stop this from happening. But then we started to think about, well, what's actually happening inside of an actual tumor? And you don't get this uniform invasion moving away, right? So it's not like you get flow out of the tumor in a ring and then all the cells move in a ring. You have places where there's no invading cells and places where there's lots of them, right? And so we started to think, could we use flow to find these invading cells? So kind of flip the script on it. And so that's what we've been trying to do for the past few years. And so we wanted to make sure that this is something that could translate into the clinic. And so we really honed in on this step of the standard of care process. So when the MRI is performed, could we take information from that MRI so that we could know where fluid is going in these tumors? And so long ago, one of my first grad students worked with a number of people to really develop an MRI method that we could do this by. And so this is the only equation, but I am an engineer, so you have to have one. I was actually told that once on a job interview, so not here. Yeah, and so basically the idea is that we use contrast agents. And so contrast agents are things that you can use to get a better signal in medical imaging. And so what we do is we deliver the contrast agent, it comes into the tumor, and then we take a series of images over time. And that contrast agent is going to move in the space. And it's going to move both by kind of just random motion as well as this directional flow motion. And so we're able to take these images and then back solve this equation so that we can understand what the fluid flow looks like. And so this is what we get from that. So we get these maps of how fast the flow is going. And this is the tumor border here. And we also get these really nice kind of vector fields where you can see where the flow is going. But how do you actually make this something that's usable and to say that we can find tumor cells with it? And that's a really hard challenge because, looking at this, it's neat, but it's really hard to tell where the hot spots might be, where the tumor cells might be. And so a couple years ago, I had a new research scientist in the lab, Jessica Cunningham, and she came in and she looked at this and she then told this story. And so in 1991, there was a cargo ship in the Pacific Ocean. And off that cargo ship fell one shipping container, and it was full of ducks. It was full of a lot of different floating things, but it burst open, and then all of these ducks at a pinpoint in the Pacific Ocean were there. And then they started to float away in the ocean. And so the people who modeled the ocean currents were very excited because they had all these models where they could track and they could start predicting where and when those ducks would arrive around the world. And so for the next decade and a half or so, people were picking up ducks off of the beaches as far away as Scotland and then reporting back to these scientists. And they found that their predictions were correct. And so she brought up this duckies story, and then it makes sense, right? Can we follow those flows to find where the tumor cells might end up? And so this is what you get when you try to do that. So these are called tumor originating path lines, but we lovingly call them duckies because that's a mouthful. And so I'll probably call them duckies here. But basically, the idea is that in the tumor, if you drop a duct or a cell or just, a pinpoint, and then you let it follow those vectors, you'll get these streamlines. And so then you can find places where you get a lot of them converging and places where you get nothing, right? Or you get none of them even leaving the tumor. And so that's a really useful tool, but then we had to show that it was actually useful. And so we could, put that onto our invading tumor with the MRI and then see how well it matches up. And so the first thing that we found, well, we found a lot of things, but just two that I'm going to highlight. So one thing that we found is that the more of these path lines that you have leaving a tumor as opposed to staying in the tumor means that you're going to have more invading cells. So those correlate. And so that makes sense, logically, but we still had to show it. And so you do see that there's more invasion in general if you have more fluid leaving the tumor. That could be very useful diagnostically, potentially, because you could think about, how aggressive you might need to be with a surgery just based on that information alone. The second thing we found, and we had to develop a different metric. So basically, if we take the duckies and then we create these kind of heat maps where there's more dense regions and less dense regions of the duckies, we're able to kind of correlate those on a spatial level. And what we find is that we get significant correlations across multiple tumors for where the invading cells are versus where you have more of these like streamlines terminating. And so that has led to, kind of us trying to make this more useful for clinicians. And so we were able to start up a company based here in Roanoke where we're trying to kind of develop software that would help clinicians, have these heat maps and better kind of define where they might want to irradiate or surgically cut. But also the other advantage of it is that not only do we hopefully find more of the invading cells, but we can maybe spare tissue that doesn't have any tumor cells in it. And then you might lead to like a better quality of life for the patients. And so that's what we're trying to work on next. So through a number of collaborations, we've been trying to look clinically. Fortunately, though, there's a lot of archival databases of imaging data that we're able to use since we kind of took a very translational approach. And so what we are doing now and what we've shown in a few of these data sets is that if you have a tumor at diagnosis, we can create these flow maps in these patient tumors as well. Then we can use that to predict where it will recur. So basically, if it's invaded and it's still there, we predict that then that's going to eventually regrow and form a new tumor. And what we found is that we're pretty good at doing that in a few, but we're still collecting data on that. But that's kind of part of the goal in validation, though we'd love to, obviously in a forward-thinking clinical study, want to do more intervention in that regard. So that's when flow is too fast. So now I'm going to switch to when flow is too slow. And so, in brain cancer, we have too much flow, but when there's some sort of cognitive issue, cognitive decline, we see that there's less flow happening. And so to think about this, we have to kind of zoom out a little bit. So I've been talking a lot at the tissue level, but I'm going to move to kind of the whole brain level and kind of the whole body level. And we really need to think about what actually controls interstitial flow, or one of the major things that controls interstitial flow, and that is the lymphatic system. So how many people have heard of the lymphatic system? Oh, good, you're well informed. Okay. So the lymphatic system is a vascular system that is really responsible for draining fluid from our body. And it does a lot of other really important things, like it maintains, tissue balance. It also helps with immunity and immune response. But if you manipulate the lymphatics, you can generally change the fluid flow inside of a tissue. So that's everywhere, right? But up until about 10 years ago, people generally said there weren't really lymphatics in the brain. No way, it's draining some other way, and that's not happening. But then a group at the University of Virginia, who I had the opportunity to work with while I was there. They kind of discovered and then had to back up and said rediscover alongside another group that there were these lymphatics that ran along outside of the brain. So those are these little green vessels here, and they're really small and they just run and they actually drain the brain. And so these vessels are really important, as you can imagine, and there's just been an explosion of research about this in the last 10 years. And so these lymphatics have been tied to cancer or have been implicated in cancer and traumatic brain injury, multiple sclerosis, infection. Kind of if you name a neurological disorder, there's probably a publication that's trying to link these together. But one thing where we hadn't seen any work being done was in something called chemobrain. And so chemobrain is a side effect of chemotherapy treatment. So basically, when individuals are undergoing chemotherapy, 75% of them experience some form of issue with memory or concentration, fatigue, and executive function. And so this is more associated with certain chemotherapies in cancer, and it's also very tied to sex and age, as well as other kind of pre-existing conditions. But this can be a very debilitating condition, and it can really, affect both the quality of life and then, just generally kind of anything that they're doing and such. So why did we want to study this? Obviously, it's an important idea, but why were we the ones to kind of get to this? So before we got into chemobrain, we had been looking at how chemotherapy actually affected the lymphatic system in breast cancer, and specifically in the breast cancer tissues, because in those tissues, if you have issues with your lymphatics, you can experience something called lymphedema, which is kind of the backup of fluid, which is also a very bad side effect of chemotherapy. And so we wanted to study how chemotherapies were actually affecting these vessels. And we had found that, at least in the two that we had studied, docetaxel and carboplatin, which are very commonly delivered chemotherapies for breast cancer, for triple negative breast cancer, you actually see changes to the lymphatics. And so we started to think, okay, well, if they're changing in this tissue, what's happening in these brain draining lymphatics? And so fortunately, Dr. Monet Roberts was in my group at the time, and she kind of took on this project to try to understand this system. And she also built a couple models alongside a graduate student in the lab. And so what did we find? So we basically took these meninges and we looked at them, and we found some really obvious changes. So one was that the lymphatics were smaller. So that means there's less surface area for fluid to actually drain. The other thing we found is that they were less branched, they were less complex, which means there's fewer entry points for drainage. So essentially, we were seeing this reduction specifically with docetaxel or taxane chemotherapy in changing these lymphatics and really damaging them. But was it actually resulting in a change in flow? And so we had built kind of our imaging methods for cancers in the brain, and here we apply it to just a normal brain. And what we're able to see is that we get this reduction in the speed of the fluid movement. So you are reducing the speed. And so that, could indicate that there might be some problems going on there, but you have to be able to test it. And so what we did is we did some memory tests, and we did this in mice. I'm trying to move that. And so basically we did a test called novel object recognition. And the idea behind this is that you have a mouse, and mice are very curious, and they will go and explore these two objects. So you put them in with the objects, they go and they explore with them. If they're both new, they spend about 50% of their time with each. And so that's what we see here in these graphs. They're spending about equal time with both of those. Now what we're going to do is give them a new object on the next day. And so they will be hopefully more excited about the new object because they already explored the other object. But they will only spend more time with that new object, so the yellow object, if their memory is working. And so what we see is that in an untreated, just normal mouse, they spend a lot more time with that new object. But in that docetaxel treated mouse, it's back to equal. So they don't remember that novel object anymore. And so here we're kind of linking that there's this flow change, there's this memory change, and there's this like anatomical change in the vessels. But so what, right? So now that we know that this is happening, what can we do about it, right? And so there's a couple things. So alongside the development of this work, we were able to develop two really cool models. So one is we generated an in vitro model, which is made up of human cells. And then we also use this cool ex vivo model where we can take full like lymphatic tissue and understand it. And what's nice about these models is now we have this tool set where we can test, how different chemos and maybe some intervention can actually, maybe cause chemobrain, but also prevent it from happening. And so that's what we're starting to do now. And not only can we do this, just with general cells, but we might be able to use kind of patient-specific cells, or maybe cells that kind of represent some of those different populations that are more prone to getting chemobrain. But we can do something else too. So in Alzheimer's disease, we had some work with the meningeal lymphatics about 8 years ago now, 7 years ago. And so this is another condition in which you see kind of loss of memory function and issues with cognitive function. And so, it's not exactly the same as chemobrain, obviously, but you're still getting some of these negative side effects. And so this is a disease that affects 6 million individuals in the US. It's higher in women. And it's marked by a number of different kind of hallmarks that we see across these patients. But what do we see with flow? So we've been working here for the past 5 years to try to understand how flow changes as we age and as mice age and as mice develop Alzheimer's disease. And so we're able to perform our imaging method. So here we're looking at a normal tissue, so there's no disease here. But even still, as the mice get older, so out to about 2 years, which is quite old for a mouse, we see that we have this decrease in the flow speed. So it's decreasing over time as we image on MRI. But if we have a mouse that now has, it's a model of Alzheimer's disease, we see that it decreases, and it decreases even more and faster. And so we get this like kind of sharp change. And so these seem like they're pretty small changes, but in a mouse that can be pretty big considering kind of what the baseline flow rates are. And so, we see these flow changes, we try to understand them, but now we want to see, the vessels that are probably implicated. And so what we see in these systems is that these vessels also shrink as the mice age. And so what we did, and what a former postdoc in the lab did, was actually develop a treatment so that we could reverse this. And so the idea here is that we can deliver this small protein called VEGF-C to these meningeal lymphatics, and then hopefully reverse these issues that we're seeing with flow and with lymphatics and possibly with memory. And so if we compare, so this is just one vessel. So this is a young mouse, and then it gets smaller in the older mouse. And then with the treatment, we get kind of this expansion of the lymphatic vessel. And so, but what really matters is, does this help, the animal remember things better? And so this time we didn't do kind of that novel object idea, but we did something else. So we did what's called a water maze. And the idea here is that you have a mouse and he's swimming around and he's trying to find this platform. And so the first time he does it, it takes a while. So it's the same as if, you were trying to go to somewhere new and maybe you followed directions, but maybe you turned at the wrong place and then maybe you had to backtrack, right? So it's going to take you maybe like 10 minutes longer to get somewhere than you were expecting to. But then the next time you do it, you should get better at it, right? You should get faster. But if you can't remember that you did it the last time, then you're going to still kind of have that slow movement, and it's going to take you still very long. But say, we treat the mouse, now he gets to the platform faster. And so that's what we see here, is that this time should slowly decrease. it should decrease every time they do this. Every time you go to the same place, they get there a little faster. And when we treat them, we actually do better than when they're not treated. And so we're able to kind of restore their memory function by targeting these vessels. And so, that's in Alzheimer's, but we hope that we can kind of take some similar strategies towards chemobrain and maybe to other types of disorders where we see similar things happening. For example, in traumatic brain injury, or maybe in stroke, where you also are having changes to flow. And so, we have this healthy brain, which is moving at a certain flow rate. And each of you probably has a very different flow rate than the person next to you, because I think it's very specific to each individual, and probably how tired you are and what you just ate and all of that. But then with a brain with cancer, we have too much flow. So there's more flow moving the tumor cells around. And we really have to account for this, both in their invasion, but also in how we treat these tumors. And then we have the other end of the spectrum where flow is too slow. And so ideally, our goal is to potentially be able to change the flow so that maybe that alone could help with these diseases, but also just really build the tools so that we know what we're dealing with and so that we can know, what the flow is actually doing. And so maybe we can't always target the flow, but we can target the things that the flow is doing or that are causing that flow. And so ideally, we all have these healthy brains with our special flow rates, and then we can really, have good cognitive function at all times. But to study these problems and to really go at it, it's really difficult. And so we have to build a lot of tools. And so I tried to highlight a few, but just to bring it back, kind of talking about the individual flows, it's really important that we are able to build patient-specific models. So incorporating cells that represent each individual so that we can really study the disease and understand, how different are the responses across different people. But then coupling that with biophysical models, where we really, use computational tools to map the flows or predict what's going to happen, and coupling that with imaging approaches. And the last thing we're really doing, and that I didn't really talk to you about much today, is that, the brain is inside of your whole body. And so it's inherently connected to everything, right? And so you really have to be able to understand how it's interfacing with those other tissue systems to explore, potentially, something as simple as how the brain goes through how things from the brain go through those lymphatics and into the draining lymph nodes. We have to build these kind of multi-organ on-chip systems, and we're doing, that in a number of diseases, including Alzheimer's and cancer. But to do that, we can't do that alone. So we have to have great people, and we have to have great partners. So I have fabulous researchers, many of whom are here. So staff and students, grad students, med students, high school students, undergrad students who have performed this work. Faculty, all of you here, as well as everywhere. And we work with a lot of different faculty, a lot of different scientists, biologists, engineers, mathematicians, physicists, chemists, to really kind of take this interdisciplinary approach to our work. We have fabulous collaborators and partners. This includes clinicians, hospitals, universities, and industry. And then our community members. So all of you want to give, a special note to, patients and their families who, take a chance and might enroll in a clinical study so that we can, understand tumors better and make better treatments. And then patient advocates. So early on in my career, I learned about chemobrain from a patient advocate. So I had never heard of it. And she had actually lost her job. Like the person she knew had actually lost their job because they had suffered from this kind of brain fog, this chemobrain. And so, that was really important for kind of shaping 10 years later, our ability to build these systems. And then we have our budding scientists. So, the young people who are coming into the field and are really important and also are really fun, and science advocates. And then last, I think Mike already mentioned, but funders, right? So the government has funded most of our work, the federal government, but also foundations and institutions. And so these are some more specific thank yous. So names of those in my lab. So Gray are those that have graduated or moved on to other positions. And then we have a wealth of collaborators. I'm sure I missed like half of them, but we love to work interdisciplinarily and really work with people across, Virginia, but also across the world. And then we've had really generous funding through the National Institutes of Health, through a lot of different institutes. And the work that I highlighted here has been funded by many of these, as well as Red Gates Foundation, Ivy Foundation, and Coulter Foundation. And then this is my family, because I promised Sadie I would put a picture in. And so with that, I'm happy to take any and all questions. You can also come visit us anytime. We're right next door. And then I'm going to put this one on. So.
We've certainly got time for a few questions. Why don't we start right with the family right here?
Oh, dear.
Get her a microphone, please, in the front row. Brad, if you can.
If you get her a microphone,
Microphone's
she might not
on,
give it
Cameron.
up.
Hold it close. Thanks.
How can chemo brain affect your social life?
Ooh. So I am not a social psychologist, but that's a good question. So chemo brain does affect people's social life because they can't remember things. They might not remember people or events, or they also, a lot of people suffering from chemo brain feel very stressed about it. And so sometimes they don't want to go engage in a public situation because they worry that they're not going to remember something. So not only does it affect their actual ability to remember things, but it also can make them very nervous to get in situations. So yeah, they can become very isolated. It's a good question.
Question right back there in the center. Can you pass the microphone back, somebody to her? Is that mic on and working, Cameron? Are you sure? I didn't hear a lot. Do we have another mic?
For that Duffy dispersion model, is that done in 2 dimensions or is that 3-dimensional?
Yeah, so what I showed there was 2D, but we do it in 3D as well now. So, but the data I showed was 2D. So first steps, and now we're working on 3D.
Thank you.
Yeah, you're welcome.
Anthony.
So in a normal situation in the brain, CSF has a particular constellation of solutes. And blood circulating is going to have a different constellation of solutes. And so given the difference in the surface area, the ventricle brain interface versus the vasculature, how does the fluid flow between the two work? Does CSF flow at a different rate? Does the solutes delivered by the brain, by the vasculature rather, and also going out as well? So lymphatic versus CSF and cortical space.
Yeah, so I think you do have kind of a pressure gradient across your brain that works alongside kind of the CSF motion by the ciliary beating of the ependema. And so you do get this movement of fluid up as far as I understand. So that would mean that like the CSF is probably coming in just a little faster than the drainage is happening to kind of keep that pressure moving. In terms of if you move into like kind of the interstitial spaces, it's like a mix of the fluids. And I know debate is still heavily going on about how mixed are they, how much are they really these separate compartments. And I don't think we fully know that yet, like in terms of like what's there. Oh yeah, so when a tumor is there, it's a bit more chaotic. Yeah. Yeah, so when a tumor is there, so one thing is that you have a lot of the flow that I'm showing here is blood flow coming in. So that's going to be fluid coming in from the blood into the space. But if you image the CSF, so if you kind of perform the same imaging, you will see like some diversions, right? But it's a balance because it really depends on how much pressure that tumor has. Yeah, but it's a really interesting question, but something we're trying to understand.
The microphone's not working, so speak as loud as you can.
Right. Great. Thank you for the talk. It was really, really enjoyable. I'm a neuroscience grad student, and I don't think about flow in the brain at all, so really edifying. I just was curious, so in my lab we study how the brain and the body interact, especially in pain. And what's like interesting to me is that a lot of different diseases have problems with the autonomic system, heart rate, blood pressure. And so like these autonomic changes feel like they're downstream consequences of lots of different things in the same way that you can, oh, I can hear myself. In the same way, like you can develop migraines for all sorts of reasons. And so what I was taking away from your talk was that fluid flow might be the downstream consequence, potentially. It might also be the cause, but
Yeah.
that's basically my question. Like what, does fluid flow changes in the brain result from things more often? Does it cause things more often? Like would you expect to find people who are born with differences in fluid flow, although you said we're all different, like they're more predisposed to be, develop cancer or something like that?
Yeah, that's a great question. It's chicken and egg problem, right? So we don't know like what causes what. We haven't gotten far enough where we can push more fluid through. I mean, it causes a host of other issues when you do that in the brain, right? Like, because that can cause hydrocephalus, and that itself is going to be a problem. So I will say that I do think it's a bit of both, because we do know that if lymphatics feel more flow, they will make more lymphatics. It makes sense, right? Like it's kind of like you need more places to drain stuff, so normally they should make more. But then that could lead to potentially long-term heightened flow, right? And so then that could lead to something else. So it's really tricky. In Alzheimer's disease, we've tried to start dissecting out, but kind of in the context of like if we deliver things like amyloid beta, do they accumulate and then do you get flow slowing? And you do see that, but then you also, as the flow slows, you get more accumulation of amyloid beta. So yeah, I think it's downstream and upstream, but I don't know. It's a really good question. And we haven't studied it at all in neurodevelopment, but others have done a little bit. But I think it's like a really fascinating thing to think about in terms of that.
Question way in the back, if you get the mic back to Brad, thanks.
When a patient's completed chemotherapy, do those symptoms start to dissipate? Do the lymphatic vessels repair at all in the meningeal area?
Yeah, that's a good question. So, patients do so about 75% of patients during chemo have chemo brain or report some symptoms. A lot of them resolve, but about 36% still report issues a couple years out with chemo brain, but some do have like kind of long-term consequences from it. In what we've done in our testing, we know at least in the breast tumor, the breast tissues, things sustain for a couple months. So you still see lymphatic changes, but we haven't actually done long-term studies in animals in the meningeal ones, but that would be a really good next step.
And then I have a really layman term question for you.
Sure, please.
So most of us who don't understand that brain flow is incredibly important, we hear the word hydrocephalic and we're like, oh, water on the brain is so bad. That's really, really bad. So what's happening with that brain, the fluid in your brain when it becomes the negative hydrocephalic?
So it's a lot of different things. So often what it is that there's some issue with either the production or the drainage of the flow, but there's different causes. So if it is going to the wrong place, things like that. And so hydrocephalus is when you're not getting enough drainage, and so it's just building up in there. And so that's why they do something like put a shunt in so they can get an extra spot to get the fluid out. So that's kind of one example. But yeah, often like if you start, the thing with the brain is it's so carefully balanced that if you start messing with the flow, it can lead to a lot of other issues. And that can be that usually you get some sort of backup and swelling because it's such a tightly controlled system.
Going through
Chemo treatment and they don't have the drainage in those lymphatic vessels there, are they more prone to hydrocephalic issues?
I don't know. I mean, there's other drainage routes as well. So that was just one that I talked about, but there's other ways that the brain drains. So it's possible that those are compensating. We see the speed slowing, but we actually haven't measured total fluid, which would kind of get to that answer, but that's something we could try. Y'all have good suggestions for grant writing, so.
Question up here, Dustin, if you got a mic to her, please.
Making him move around.
I'm assuming that the pressure gradient in these glioblastoma tumors is greater than in other tumors, or because that's why because of the invasiveness that you're talking about. Does this pressure gradient also exist in other cancerous tumors, breast cancer, yeah, if you could just talk about that.
Yes, so it's actually not greater in GBM than other tumors. It's about on par. Sometimes it's lower, but yeah, it was actually this phenomenon of having this increased flow with this pressure differential was described it's been described in melanoma, in breast cancer. It was one of the main things because at the time when this was first being studied, that was one of the big research initiatives. And so, yeah, it's kind of seen across multiple tumors. We've done the same type of imaging in pancreatic cancer, in prostate cancer, in breast cancer, and it's happening in most cancers. But what we don't know is how tightly it's connected to this invasion. In breast cancer, we know that it is. In melanoma, we know that as well. But we don't know that that's universal or if there are potentially other consequences.
One more question over here.
Can I just ask a little follow-up? But
Yeah.
what is it, because I was hearing from your talk that pressure gradient causes that invasion of the surrounding tissues, which makes resection very difficult.
Yes.
But you think about in breast cancer, what I know of breast cancer, it's a little bit more successful to have resection. So
Yes.
why in glioblastoma is it more difficult than in breast cancer?
So I think the tissue sparing in glioblastoma is a major thing, right? I mean, you can't do a full mastectomy of your brain type of situation. So that's one thing. And as I mentioned, there is a bit of a connection between fluid flow and invasion, but there's also this whole metastatic cascade too that happens with breast cancer. So if those cells are invading into the tissue and they just remove a lot more tissue, and we have better, I think, some ways that they plan those surgeries. So in GBM, they often, the patients really get the same type of treatment. Breast cancer has become much more specialized, right, and much more patient-specific already. And so I think because you have this metastasis component, sure, they invade the tissue, maybe remove the tissue, and then if it hasn't metastasized, that should work pretty well. But brain cancers don't metastasize outside of the brain. They stay in the brain, and so they're going to be in the brain somewhere, and we can't remove it.
Is there a question?
Hi,
If not-
thank you.
If not, Brad.
Oh,
I
she-
think-
Yeah, I've got
Yeah.
the mic.
There she
Yeah,
is.
she's got
Go
it.
ahead.
Thanks for your talk. So I was just wondering, so you mentioned the increased flow, it's going through the invasive zone. Aside from the invading cells obviously being an issue, does the increased flow, have you observed it damaging the healthy tissues that it's going through, or is it just sufficient to drive the chemotaxis that you were talking about?
Yeah, it changes a lot of things in the tissue. And so I didn't talk about them, but one of our patient-specific models includes the other brain cell types as well. So you do see kind of the cells become, the astrocytes, microglia, those that are there, becoming potentially reactive in those regions. But one of the things we're working on right now is trying to apply our flow imaging to map on all of these different microenvironmental factors. In vitro, like in a dish, I don't know about damage. I can't remember where we've seen potentially viability issues, like cells dying, but you definitely see cells changing. So potentially you do that long enough, they might die. Yeah.
Okay, last question right here.
Thank you so much for your talk.
Yeah.
I work in an exercise lab, so I'm tempted to think that exercise is good for everything.
Yeah.
But I was thinking about if increased flow may be contributing to this invasion. Is the increased flow that you see during exercise potentially exacerbating the invasion? And then sort of tied up with that is the decreased flow that you see with chemo potentially protective against this invasion?
Yeah, so good question. So I will say, I was showing, kind of in a vacuum, these are tumors, they're growing in place, they're not undergoing any treatment. In patients, it's a lot more complicated. Like the higher flows that we see in patients don't necessarily mean that they have worse survival. And I think that part of that is, flow, I'm linking it to invasion here, but it's these kind of, the movement of fluid is also incredibly important for getting drugs in, getting oxygen in, which makes radiation therapy work better. So I do think there's a balance between, how bad is it on the invasion at the initial stage versus once you start treating, these heightened flows can be really good for getting the drugs there. Plus they have exercise has a host of other benefits for cancer patients in terms of, things that really contribute to overall survival. So no, they should not stop exercising if they're able and can do it safely. With the chemo brain, yeah, it's possible. When we first started studying that, we were actually so in breast cancer, we see more lymphatics. And so we thought we would also see more lymphatics with the chemo treatment. And so we were actually exploring it as a potential treatment for Alzheimer's disease or something where you see reduction. But then we actually saw the opposite effect in those lymphatics. So in a different tissue, we were seeing an opposite effect. So I guess that's to say, I don't know. I think the body works in really strange ways. But it could be an interesting thing to explore in the context of exercise, definitely.
Before I end, I want to say I just lied. There's one more question, and I'm going to ask it.
I know.
20 to 30% of your patients that didn't have chemo brain, what about the lymphatics in those folks? What has anybody seen or have they looked?
I don't know.
Okay.
So we don't know. So we haven't looked at these in patients yet. I mean, obviously, they're not easy vessels to get. We are trying to start some studies looking at radiation therapy. And also, I think the one thing we can really do is kind of combine our glioma work with the chemo work and look at these vessels in, patients with brain tumors who are getting chemo. Because in those cases, when they get surgery, you can actually look at these vessels. So we can't do that in a breast cancer patient.
Please join me in thanking Dr. Munson for answering.